Good afternoon. I'm sure you're all exhausted by now. I want to thank the Foundation for inviting me to come and speak. And I'm going to talk to you a little bit about rapid onset dystonia Parkinsonism, which is another ATP1A3 disease. So what is rapid onset dystonia Parkinsonism? It's a rare disease. It's actually more rare than AHC, if you can believe it. It has an age of onset range 6 months or a year to 55 years. It's inherited as an autosomal dominant trait, which means you only have to have one parent who passes the gene on. You only need one copy of the gene. There's reduced penetrance, which means you can have the mutation, but you don't necessarily show the clinical symptoms. But there's also de novo mutations, which is what the vast majority of AHC families encounter are de novo mutations. The symptoms are acute onset, hours to days, of dystonia and or Parkinsonism with little or no progression, and it usually is triggered by something: physical exertion, emotional stress, heat. There's some slight benefit from L-DOPA, but not much, and mutations in the same gene that causes AHC also cause this disease. So this is so when we found the gene for this disease, it was in 2004, and so this was before the Human Genome Project, before we had exome sequencing, and so what we used back then were large families, and so this is a large family that has this disorder. So unlike what you guys generally you don't have any other person in your family with the same disease as you. Here we can see everybody who's colored green has this disease. And the reduced penetrance, you can see here, for instance, here's a person who's affected, but their parent is not affected. So that parent has the mutation, but they're not showing any of the clinical signs. So we had 12 affected individuals, and they had onset of symptoms minutes after childbirth, while giving a speech, running, after playing basketball. So triggers. The age of onset in this family was from 14 years to 43 years, and it usually stabilized 1 day to 60 days after onset. So this is called rapid onset dystonia Parkinsonism because basically these people go out jogging, they come back from jogging, and suddenly they get this movement disorder, they get stuck in these positions, and that's it. They're just stuck like that for the rest of their lives. So unlike what you see in your kids where you have this paroxysmal, they have their attacks, but they go away after a certain amount of time, and they have multiple attacks, these guys have one sort of attack-like event, and they get stuck. So this is just showing you the proband, the first person that came to clinical attention in the U.S. in that large family. And then we found another family in Ireland. And this is the proband from Ireland. And you can see they actually look quite similar. And they basically have dystonic spasms of their face. They have masked faces, which is what you see in people with Parkinson's disease. They have drooling, this gaping mouth. They're unable to eat or swallow. So you have some of these same symptoms in your kids. And this first U.S. proband actually was described in 1992 and came to Riley Hospital here in Indiana. So this is the first time that RDP was ever described here in Indiana, actually. So these are different mutations in the ATP1A3 gene that cause rapid onset dystonia Parkinsonism. And basically over here is a list of clinicians that have sent patients to us in order to find these mutations. And these clinicians are basically from all over the world, from Europe, Asia, the US, Canada, and even South America. So these patients are from all over the world. And also, like you find in AHC patients, there's a particular mutation that is overrepresented or found more often in these families, among these families. So there's a hot spot, just like you have several mutations that are hot spots. So what are the diagnostic criteria for RDP? So in the majority of cases, there are exceptions, but in the majority of cases, you have this abrupt onset of dystonia with Parkinsonian features. So they have this rigidity, this stiffness, this slowness, and it occurs over minutes to 30 days. They have what's called a rostral caudal gradient, which means they start usually in the face and move down the rest of the body, which is opposite of other forms of dystonia, which usually start in the leg and move up. They also have prominent bulbar symptoms, so that's the swallowing and the drooling and things like that. Other common features are this report of triggers, that they usually stabilize within a month, that sometimes they have a rare abrupt worsening, so they could have another event that could be triggered by something else that makes them worse. They have occasional mild limb dystonia prior to the RDP, so they might have some trouble writing or something like that that comes and goes. So it's a little bit like the paroxysmal nature that you guys have, but it's not seen in all patients. And overall, there's minimal improvement. And the additional features, they do have some seizures, not as common as in AHC kids. They have psychosis and they have cognitive impairment, but not as bad as in AHC kids. And so this is just some of the faces and families with RDP. And so this is a young lady from Ireland. These guys are from Poland, a family from Poland. This is actually what would be the great aunts and uncles and grandparents of that child that came here in Indiana. And so this is that big family that we used initially to find the gene. And these are two other families that are here in the US. So, of course, you guys know all about alternating hemiplegia of childhood, and that the gene was identified in 2012. And these are just showing the different mutations, and we've gone over this with multiple speakers. And here's the different mutations that are recurrent in your guys, as well as this G947R. So you also have these recurrent mutations. And of course, this is the diagnostic criteria for AHC, which you guys know very well and have seen for the last day or so. So now this is a comparison of AHC and RDP, the clinical features. So here you can see that AHC, the age of onset, of course, is very young, before 18 months, and RDP is older age of onset. And both of them have triggers. Some of them are similar triggers, some of them are different triggers. Both of them have many of the same neurologic features: dystonia, dysarthria, drooling, this rostral-caudal gradient, slow, unsteady, ataxic gait, some bradykinesia. But you guys have the hemi and quadriplegic episodes that you don't really see in RDP. There's some seizures in both. And then these other ones, choreocystosis, developmental delay, abnormal ocular movements, we don't really see that much in RDP, though there is some developmental delay, but it's more like learning disability. So these guys go to school, but then they have some trouble in school. And of course, we also have this abrupt onset. You guys have the paroxysmal nature of this abrupt onset, and then these are slight differences in that you're paroxysmal, and once this starts, it pretty much stays. So I wanted to point out that, in fact, there's now a whole group of diseases that are caused by mutations in the same gene. So in addition to RDP and AHC, we have a whole bunch of other diseases now. All right? So I'll try to talk into this still. So we have this EIEE, which is early infantile epileptic encephalopathy. Here's AHC. We have CAPOS, which is cerebellar ataxia, areflexia, pes cavus, optic atrophy, sensory neuronal hearing loss. We have RECA, which is relapsing encephalopathy, cerebellar ataxia, and here's RDP. In addition, there's other phenotypes that are not on here. For instance, I know of a case of a young onset ataxia patient, which is mainly, that's all they have is ataxia, but it was a sudden onset of ataxia, which is not often seen. And so this shows you the different onsets that go with the different diseases, and it shows you the symptoms that span the different diseases, and it shows the frequency of these different diseases and when they're triggered. And so you can see it's almost as if there's a gradient of symptomatology, age of onset, and it's related to particular mutations as well in this same gene. And so as exome sequencing continues and comes on, we're going to probably see lots more potentially diseases that are actually related to each other, and you know, we may start calling these ATP-related diseases or ATPopathy. I don't know what we're going to call them, but we can't necessarily keep calling them what we're calling them, because now they're kind of this whole group of related diseases that we have to start thinking about and looking at the different kinds of features that each one has, and that's telling us things related to the mutations in the gene and how the gene is functioning and things like that. So this is just showing you all these different mutations that are in this gene, and the blue are the RDP mutations, the red are the AHC mutations. There's a couple of red with blue centers. Those are mutations that are found in both AHC and RDP. And I think there's a purple. I think it's right in here someplace. Yeah, right there. That is the Capos mutation. So there's, for this disease Capos, there's only one mutation, and there's about 7 families now. They all have the exact same mutation. And so in general, the mutations in AHC severely reduce the enzymatic activity, whereas the mutations in RDP only reduce the protein levels without affecting the enzymatic activity. And so it's thought that the RDP mutations are less severe than the AHC mutations, which is why you're seeing this different phenotype, why you're getting the later age of onset. And you know, maybe the difference in cognitive impairment is because you're limiting the amount of this enzyme really early on in AHC versus later in development in RDP, something like that. And this is just showing you this difference between protein level and protein activity. So up here you have the AHC mutations, and here you have RDP mutations. So DYT12 is another name for RDP. And this is showing you the protein level, and you can see here's this WT we talked about earlier. This is wild type. You can see that all of these guys have, you know, within reason, the same amount as wild type. Whereas with the RDP mutations, many of them, not all of them, but many of them have very reduced amounts of protein. But if you look at the activity, here's the wild type again. So all of these mutations pretty much have lower activity, but the DYT12 or RDP mutations have higher activity than the AHCs. And so this probably translates into the somewhat the difference in the phenotypes that we're seeing. I don't think it's going to work. I got this movie from Dr. Swender at Mass General, but it's not going to show it to us. It's basically showing you this is the membrane, and this is the actual pump in 3D, and it's showing you the, oh, here we go. That's the sodiums going in, and here's the potassiums. So this is showing you the sodium and potassium switching, and down here is the ATP that changes from ATP to ADP, and you get this conformational change, and that's how the molecule works within the membrane. This is how the pump works within the membrane. And so I thought it was cool, so I thought I'd show you. And it's much better than anybody else showed you so far, right? Come on. Give it up for Kathy Swender. And so this is also from Kathy Swender's work. And so this is very interesting. So what she's done now is she's mapped all of the ATP1A3 mutations onto the structure. And so you can see that there's actually clustering of these mutations. And now when you divide them into severe symptoms, so AHC-type symptoms or other symptoms happening in infancy, versus milder symptoms like the RDPs and like that ataxia, that kid with ataxia, you can see again that the mutations aren't really overlapping. So they're in different places. So although when we looked at the linear structure, you could see that the mutations were sort of near each other and overlapping, when you now put the mutations into the protein structure and put it in 3D, and the way it folds up, you can see now that the AHC mutations are clustering and the RDP mutations are clustering in different places. And so again, this is helping us to explain or understand the phenotypic consequences of, why is the phenotype different between AHC and RDP and these other diseases. So somehow it's affecting different places. So for instance, right here was where we saw that exchange of the sodium potassium, and we got a whole bunch of these AHC mutations on top of that spot. So it's probably somehow mucking up that exchange of the sodium potassium. Whereas the RDP mutations are down here near the ATP, where the ATP is switching. And so maybe, that's somehow involved in what the RDP is doing. And remember, we're not seeing that conformational switch that we saw in the movie, so that when the sodium and potassium exchanges and the ATP exchanges, you get these rotations that then bring these molecules into different alignments with each other again, or these mutations into different alignments with each other. And so all of that comes into play with how, whether these are severe or mild. So I'm going to switch gears slightly and talk about a study that they're doing, my collaborators are doing down at Wake Forest, and I think some of you probably ran into them on Tuesday and Wednesday because they were here when you guys were having your patient evaluations. And so Dr. Allison Brashear is down at Wake Forest, and we have a grant from NIH to study RDP and AHC, and one of the aspects of it is to do imaging. And so ATP1A3 is highly expressed in these regions of the brain, which I think you heard about a little bit earlier today. It'd be good if I could spell. The hypothesis is that RDP may affect pathways associated with other dystonias. So rapid onset dystonia Parkinsonism is just one member of a group of diseases called dystonias, and the dystonias are related to different pathways in the brain. So this corticostriatal pallido-thalamic cortical pathway and this cerebellar thalamal cortical pathway. So these are just pathways in the brain, and the question is RDP related to some of these same pathways that we think are involved in other forms of dystonia? And so they're going to use MRI and DTI to look at this. And so the other thing that we have, so 4 of those people that were in that picture that old picture from that original family, 4 of those people have died and given their brains for autopsy. And so we've been doing some studying of those brains. And if we actually look at what we see on the imaging, we see some differences. So the yellow are the differences that we're seeing between RDP patients versus controls. And there's particular regions of the brain, so this cerebellar region of the brain and this dentate nucleus region of the brain that are showing differences. And if you go and you look in the pathological samples that we have, you can actually see when you look in the cerebellum or the dentate nucleus, you can see this neuronal loss. So for instance, this is a person who's affected versus a person who's a control, and you can see that this kind of brown staining you can see very well in the control, but you can't really see it at all in the affected. So that suggests that what the neurons that are staining brown here are missing. And you can see here also, there's these are also neurons. And you can see how nice the staining is here, and here it's much lighter, suggesting that we're losing neurons here. And again, you can see the difference in density between, and here, for instance, you can see these sort of purple circles, and there's not so many in this slide. So it suggests that we're having some neuronal loss, actually, in these brains. And that neuronal loss is actually correlating with some of these imaging changes that we're seeing. And so we really like to do imaging. And so what they're trying to do at Wake Forest is actually now ask, so the average age of onset in RDP is maybe in their teen years or 20s, whereas, and so the question would be, are there AHC kids who have reached their teens or their 20s, and can we compare the imaging and ask, do their brains look similar or different to RDP patients, because we know that they have mutations in the same gene. And so would we be able to compare the two and see, are they the same or different? And so that's basically what the study is to look for. And so, oops, I have to just write. So it's basically a 2-day protocol where they review the patient history, they do consent, they do a neurologic exam, a neuropsychological assessment, a cognitive assessment, and then they do the MRI, which is about a 45 to 60-minute time period in the scanner. So they've currently scanned 5 RDP patients, which is what we saw the data from, and 1 AHC patient, and they have another 6 RDP patients scheduled, and they have 2 other AHC patients enrolled. And so it would be really great if we could get some more AHC patients to go down to Wake Forest and think about doing this MRI and this 2-day protocol. And then hopefully we can get some answers. Is AHC and RDP similar or different when we look at the brains? And I'm going to now turn it over to Dr. Swoboda. Can we just have questions at the end?
Is there any cost involved in going down to Wake Forest and getting this done?
No, the costs are all paid for by the grant.
Oh, thank you.
No, unfortunately. I think Wake Forest is in the middle of the state.
Question here.
On the slide where you were showing the 3D model of the brain where the AHC mutations were grouped, and it seemed like there were 3 colors. Did those 3 colors represent the 3 most diagnosed mutations?
No. So I'm trying to remember which way it was. I'm going the wrong way. They're just different. Each one of the balls represents different mutations. So you can see here there's like a whole ton of different mutations. Like all of these represent different mutations. And so those are translated then onto the 3D structure. And so all of these represent different mutations. So within here are your 3 most common mutations, but I can't tell you exactly where they are. Kathy could probably tell you exactly where they are, but I can't. And so I don't know exactly. I think the colors are related to where they are on the linear molecule. Which slide? Which slide? This slide? Yeah, well, luckily you have kids. Yeah, so what happens is some of the kids who have RDP, they go out and they've had alcohol binges, and then they have a second worsening and they get much worse and they end up in a wheelchair and really bad. And so actually Allison now talks to the families about like avoiding alcohol. Yes, it can happen. Whoever, if you have your first episode, usually this is a second episode when they go out on binge drink or something like that. They drink a lot of alcohol. It's not like you have one drink. It's like, So yes, alcohol is a trigger for RDP.
On the slide with related diseases,
Right.
What is the gravity actually tracking?
What is the what?
Gravity?
Severity, yeah.
Third from the bottom?
This
on the
one.
left? Oh.
So this is the most severe. These are the least severe.
Oh, I see.
Severity. This is from a European paper. They don't know how to speak English. They come up with things like gravity.
Anybody else?
Here.
I have one more question. That was on the Parkinson side, right? So if someone had Parkinson's, drank too much alcohol, that RDP thing could happen. That's it. That's what my question.
That's on the RDP.
Yes.
I figure your kids who are only, 18 months aren't drinking alcohol, so we don't have that test.